Now, we were talking about the plant's reproduction in flowers yesterday, and we said that the flower has different structures that are some are supportive and some are structures that are reproductive structures, direct reproductive structures. Now, the we don't need to talk much about those structures which are supportive in reproduction. Like they are supporting, but they are just, you can say, um, supportive structures. So we don't discuss those structures here. We discuss the structures that are really reproductive structures. So we have that male part here, and the male part overall was called as the stamen. Overall, its name was stamen, and the stamen has had the filament, and I clearly told you the purpose of the filament in the previous lesson. Now, anther. Anther basically is the organ where sperms are made. Sperms are made. You can say that in animals, the sperms are made in the testicles, while in the plants, the sperms are made in the testicles. In the anther. So the anther is really the crucial organ. If obviously a plant cell, a flower is a male, and if it is losing its anther, then it means that it would not be reproducing anymore. It would not be reproducing anymore. So anther, but the problem is when the sperms are made in the anther, they need to be transported. They need to be transported. And if the anther, if the sperms need to be transported, where they need to be transported? To the egg. Now, if they need to be transported to the egg, you must have a of transport. Remember that there was a time when the plants used to use water to release the sperm, and the sperm would just travel through the water, entering into the egg. But that phenomena is working no more. That phenomena is working no more. The plants are no more dependent on water. So, what does it mean? It means that we need to find another way. So what do? We pack the sperm, these plants pack the sperm in a microscopic flying structure, flying structure called as what? Pollen. So basically in anther, you would find different compartments which are called as pollen sacs. The design of anther, if you just take a transverse section of the anther, you would find the anther in arranged in eight shape like this, and the anthers have particularly surrounded portions called as pollen sacs. These pollen sacs are just like, you can say, the pollen sacs are surrounded by the membranes which typically are they look like the cell membranes, although they are not, but they look like the cell membranes. So pollen sacs are particular surrounded regions in which the pollen grains are made. The pollen grains or pollens are made. These eight shaped things are called as anther lobes. You can say the outermost part is the anther lobe. The inner part is the pollen sac, and inside of it you would find the small pollens. These are pollens or pollen grains. Now the pollen grains. Remember that the pollen grains, obviously I have said that the pollen grains are flying microscopic particles. But the pollen grains contain the sperms, but not directly the sperms. Remember, the pollen grains are actually unicellular structures. They are unicellular structures containing one nucleus that is going to code for a pollen tube. Okay, what is a pollen tube? Basically, we'll be talking a bit more about it, but remember that a pollen tube is a structure that would lead the sperms, that would lead the sperms to the egg, to the egg through style. Through style. The style is the part of female reproductive part of plant. If you see, this is the style. So basically, the pollen tube nucleus is going to make the pollen tube, which would be leading the sperm to the egg, and generative nucleus. Generative nucleus is a nucleus that would be divide into two, divide into two sperms. When it is close to, when it is close to egg. So you can say that the pollen grain itself is not a sperm. The pollen grain itself is not a sperm. It is not containing the sperm directly. It is containing the equipment to make the sperms. And when the pollen grain would reach the female part, it would make a pollen tube that would take the sperms to the egg. And how the sperms would be produced when the pollen grain would fall on the female parts of the plant, then of the flower, then the generative nucleus would turn into the sperms. Is that clear?
Yes.
Now, next part is the uh, female part, female reproductive part of the flowers. This female reproductive part of the flower contains many different parts. Some are not to be discussed here. Labelings are extra. So I'll explain what not to be discussed here. First part is the stigma. The stigma basically is a sticky and sugary part. Okay, what would be the purpose of stickiness of stigma?
So that it can, uh, s- what do you call it, stick with the male part of the flower.
Actually, let's make it more simpler. When the pollen grain is coming with the air, it's coming through the air, it would fall on the stigma. The phenomena called as pollination. Now the pollen grain must not be leaving it. It would be considered as the successful pollination if the pollen grain would not leave it. And to not let it leave, we make the stigma stickier so that it stick the pollen grain to it. And why the sugary? Remember that to build a pollen tube, to build a pollen tube, the uh, pollen grain, the pollen grain performs respiration. What is the result of respiration? Energy.
Yeah.
To perform respiration, the pollen grain requires sugar. Requires sugar. So that is basically the reason the sugar would be present there on the stigma. Clear?
Yeah.
Now I told you the purpose of the style. The style is there to see the to check the fitness of the pollen grain. Because remember that the pollen tube would be made through the style. So if the pollen tube is not fit enough, If the pollen tube is not fit enough, it would be not able to pass through the style to reach the egg, and obviously that would not be a good news. So that kind of genetic material no plant would want. See, this is the way of the nature's work. The nature work for the fittest organism. If the organism is fitter, then it would be passing the test. Passing through the style is the test for the pollen grains. If the pollen tube is made effectively, then the genetic material is useful. If it is not able to do that, then it is not fit. Just give me a few seconds. Okay, so passing through the style, the pollen tube is basically supposed to be reaching the egg. But remember that the last part, which I mentioned, that why we name it ovary, because ova or ovum refers to the egg. Remember that ovary does not contain the egg directly. It contains a protective shell called as ovule. Ovule. Okay, now you need to be very, very much attentive here. After fertilization, after fertilization, the ovary would become the fruit. Ovary would become the fruit after fertilization. What is the meaning of it? When the reproduction is successful, now the ovary is going to be the fruit. The pulp that you eat, the pulp of a fruit that you eat is actually the cells of the ovary. And ovule after fertilization becomes the seed coat. This is very important to understand, that actually when the fertilization occurs, the structure of the whole plant is revised. The structure of the ovary is revised. Once the fertilization is done, the you will see that ovary has turned into the fruit, and the ovule has turned into the seed. So remember that the fertilization, and when the fertilization would occur, we obviously know that when the sperm reaches the egg and fuses with it, that would be the fertilization phenomena. And the fertilization phenomena changes the things. It would be changing the ovary into the fruit, and the ovule into the seed coat, and the egg, which is ovum here, this is the egg. This is the egg. And one more thing we need to know here, that is definitive nucleus. This definitive nucleus is very important, but we will not discuss this nucleus here. So obviously we need to know that what are the labels. Did you understand all this?
Yes.
In the conversion of ovary and ovule is very important to understand, and also where the egg is placed. First the ovary, then ovule, and then the egg. And the definitive nucleus placed close to the egg is also very important. Okay, you do not need to um, study about this slide. This is not in the syllabus. Also, the funicle, the placenta, these are not to discuss. The ovary wall is fine. So when I'll send it to you, please take care of the things that are not now required in the syllabus. This is the part of the previous syllabus. Okay. Now, what is the pollination? Read the statement here. The pollination is a transfer of pollen from stigma to From anther to stigma. Anther is the point where the pollens are made, where the sperms are made, and they are packed into the pollen. And stigma is that sugary and sticky part of the female reproductive system. Now, it is very important to know that the travel from okay, the travel from anther to stigma, anther to stigma is risky based on many factors. But remember that a flower having having both male and female parts, both male and female parts has uh, most chances of pollination. It contains maximum chances of pollination. Why? See, what are the risks? We don't know. We are not here to talk about the risks. But remember, when there is when the pollen is coming, there might be a change in the direction of wind. If an insect is bringing the pollen, maybe that insect has been hunted. So it means the pollen is useless now. The pollen is useless now, which means that there are the flowers which contain both male and female parts on a single flower. Those flowers are called as bisexual, just like this. And then there are flowers which are containing only male or only female parts. Two, that they would contain only male or female parts. Now these flowers are called as unisexual flowers. Okay, now one more thing. Be very attentive. There is another point, very important, that there is another very important point, and that is when the flowers are pollinating, there are two ways. One way is if the pollen comes out of anther and fall on the stigma, and the parent plant remains same. The plant remains same. Then this kind of pollination would be called as self-pollination. That the plant is pollinating itself. The plant is pollinating itself. Let me repeat it. When the anther is releasing the pollen, when the anther is releasing the pollen and the pollen is falling on the stigma of the same flower, or the flower of the same plant. See, this stem represent that the plant is still the same. So it means that this kind of pollination is going to be called as the self-pollination. The main plant is same. It has two cases. One, when the pollen uh, comes from the stigma, from the anther to the stigma of the same flower, that is self. If it is released from the anther but to the stigma of another flower, but the plant is same, still it would be the self-pollination. Then comes the second type of pollination, which is a cross pollination. Cross pollination basically means that when the anther is releasing the pollen and the pollen goes from to another plant. See, this is plant A and this is plant B. Pollen goes to the stigma of another plant. but the species should be same. Species should be same. So that would be called as cross pollination. That would be called as cross pollination. Is that clear?
Yes.
Okay. Now there are advantages and disadvantages of self and cross pollination. So let's see. Advantage of first of all, okay, this is for self pollination. So let's see. Uh, for self pollination, only one parent plant is required, and the offsprings inherit same qualities. That is sometimes very beneficial. That is something sometime very beneficial, that offspring would inherit the same qualities as the parent plant. Why the same qualities? Because no matter how many times you perform the meiosis, because we know that the meiosis bring the changes. If you remember, I told you that when the meiosis occurs, the gametes have different genetic material. Remember that?
Yes, I remember.
So it means that when the meiosis happens, the genetic material would be different. And now you can say that, but the problem is when the parent plant is same, when the parent plant is not changing, it typically means that you cannot change the genetic material that much, because ultimately the things are repeating again and again and again and again. They are being repeated. So if they are being repeated, that clearly means that no matter how many times you perform the meiosis, no matter how many times you perform the cell division, no matter how many times you perform the reproduction, the genetic material would remain same, because the parent plant is same. So in this case, the good qualities of the parent plant would be transported to the next generation. That is very good. That is very good. Another advantage is the self pollination does not depend on external factors, just like wind or insects. That is the problem with the cross pollination. It becomes riskier. It becomes riskier. Why it becomes riskier? Because we know that very well, that when we have wind or insect dependence, sometime the wind is stopped, or maybe the insect is hunted, so the pollen would be wasted. So in the self pollination, you can see the fourth and fifth points, that the pollination is most likely to occur when there is self pollination. It is most likely to occur. And fifth point is that less pollens are wasted. That is just because they are performing the pollination on the same plant. But when it comes to a disadvantage, one disadvantage is, these two disadvantages that are mentioned, they are heavier than all the five advantages mentioned there. The genes would remain same. Less variants are there. Continued self-pollination may lead to offspring more weaker, smaller, and less resistant. Why? See, it is a very genuine phenomena that where there is a chance of accumulation of fitter genes, there is an equal probability of accumulation of less fitter genes, which are going to which are going to plant, which are going to make the plant less resilient to the nature, more vulnerable. That is why it has this disadvantage cannot be replaced with the advantages. This is something very serious. Any question?
No.
Now, advantages and disadvantages of cross pollination. Let's see the disadvantages of the cross pollination first. Two parent plants are required. Obviously, that is also a problem. That when the two parent plants are required, then there is a difficulty. Maybe the distance. Obviously, maybe there is a difficulty of the uh, um, pollination success in pollination. They are being hunted. Maybe they are being the wind is stopped, so the pollens are wasted. There is lower probability. But going to the advantages, the biggest advantage. on the basis of what most plants are right now surviving. And that biggest advantage is when you cross between two different individuals, between two different plants, there is a very high chance that both are both are surviving in nature. Obviously, if they both are surviving in nature, their good genes would accumulate, and there is a bit lesser probability that the bad genes would accumulate. A bit lesser probability. So they would be more resilient to nature, less vulnerable, more surviving plants. So that is a very good thing related to the advantages of cross pollination. So greater variations increase the chances of species surviving. Then seeds are capable of surviving. Now this is one of the very important point that is also called as seed dormancy. This is something very strange. A dormancy is a phase of no metabolism, but the organism yet the organism still lives. Organism still lives. And in this case, the some seeds are capable of surviving for 20 years and more. This is the advantage of seed dormancy. So that we need to know, we need to discuss, we need to find out that the seed dormancy. Okay, when you try to sow some seeds in your garden, what do? Do you go and take the seeds out of the fruits, or do you go and buy the seeds from the market?
You buy the seeds from the market.
Yes, we buy the seeds from the market. And when we buy the seeds from the market, those seeds are not fresh. They are dormant. They are same as fresh, but they are not uh, just taken out of the fruits. They are dormant and they would be equally good. All right. So, as we said that the self-pollination does not require any of the factor, but the cross-pollination does require different factors to be successful. One is called as wind pollination. One type of cross-pollination is called as wind pollination, and the other type of cross-pollination is called as insect pollination. And remember, the flowers even look different for flowers look totally different. Both kind of flowers look totally different. Now, what does that mean? Let's see. Let's see first the flower that is the insect pollinated. Remember, there are few features. They have to be very brightly colored so that the insect can see them. Brightly colored petals. Second, they have to have a nectary, and that nectary is always below the ovary. And they have scent, very sharp, very sharp scent, so that the insect and birds are attracted to them. Remember all these three features. Another very important feature, their stigma is deep inside. Stigma is deep inside the flower, so that insects can insects go deeper. Insects go deeper. So these four are very important points of an insect pollinated flower. First of all, you'll see these pictures are not as good. Let me scroll down. Look at these pictures, this one and this one. You can clearly see the brightly colored, very beautiful flower. Very beautiful. And you can see that now. If you want to know, this flower never looks like this. This flower never looks like this when the insects see them. Okay, let me try finding out how the insects see them. That would be very much interesting for you. How do insects? Uh, insect- okay, hold on. Let me show you. Some pictures are shown here that look exactly like this. This one. If you see this one and this one, they have a very clear difference of colors. The insect would always look at these flowers at their sharpest. The outer large petal, this petal is named as the wing petal. The large petal is known as the wing petal. I'm sorry, the large petal is called as the standard petal. And the petal inside of it, this one, these are called as the wing petals. Still, you cannot find the stigma, or even you cannot find the answer. Okay, and then if you open the, if you pluck the wing petals, when you pluck the wing petals, you will see a nectar guide. Nectar guide means this would guide that, okay, nectar is here, come. So the insects would follow this nectar guide. Inside of the wing petal, you would find more petals called as keel petals. You cannot see these keel petals from outside. These keel petals are situated inside the wing petals. So you see, the insect would have to make through. The insect would have to make through the make through the flower petals. Like it's not that simple. And in the meantime, the struggle, the struggle would lead to struggle would lead to uh, either pollens sticking to insect, sticking to insect, or brushed away, brushed away from the insect. That is the benefit of the struggle. When it would make through, it would have to struggle, and the struggle would cause either the pollen to fall on the insect, or they would be picked out of the insect. That is the important part in the struggle. All right, is all this understandable?
Yes.
Then moving a bit more inside the keel petals, then you would find that the keel petals are hiding this particular stamen trough. And why we are saying that this is a stamen trough, this is very strange thing, because in this stamen trough you would find the stamen that is a male part, as well as the stigma that is a female part. So all these are stamens. You can say the thicker part are anthers. These are anthers, and over here somewhere you would find the stigma, the female part. The female part, although has the sticky end, but one more thing you would find here, and that is brush, the brush end. This brush would take the pollens away, take the pollens away from the insect, and the pollens would make through the style and reach the ovules to fertilize the egg. Fine?
Yeah. Mm. Yeah, understand.
Is it fine?
No.
Okay. This is how they are showing that insect would enter. And always remember that this insect is moving around here to reach the nectary basically. All the insects are actually targeting the nectar. So nectary is their final destination. Moving ahead, the wind pollinated flower. Wind pollinated flowers have no brightly colored petals because they don't need the insects. No brightly colored petals. They got no scent, and also what I told you there, no nectary, no nectary, no nectary, and no. Oh, sorry. Stigma and style, stamen, or I would say stamen and carpel are high in the air. Why they would high in the air? To capture the pollen, or to release it. To capture the pollen, also or to release it. The stamen and carpel, like male and female parts, have to be high in the air. Got it?
Yeah.
Okay. A very important thing. A very important thing is that the wind pollinated flowers, the wind pollinated flowers, they are their sepals and their petals, they are not named as sepals and petals. Instead, the sepals are called as non-flowering bracts, and the petals are called as flowering bracts. Like their sepals and petals are basically called as bracts. When they are sepals, they would be non-flowering, and when there are petals, they would be flowering. So remember that wind pollinated flowers, when we try studying them, then the wind pollinated flowers also don't have the don't did not get the name of sepals or petals by the scientists who named them. Why? Because typically these sepals and petals were named for those flowers which are brightly colored, and they have a good scent. But when it came to the those flowers which are like grassy flowers, these flowers did not get the name of sepals and petals because they are not brightly colored, they are not good scented, they are not as much conspicuous as the insect pollinating flower. So that is why we did not give them the name of sepal and petals, we named them bracts. The sepals are called as non-flowering. Non-flowering basically means that they would only cover the flower, just like I said, the sepals cover the bud, so they also cover the flower bud. The flowering bracts are those which contain male and female parts. Male and female parts. That is why we call them flowering bracts. Understood?
Yes.
Just hold on a second. Okay, now just look at this flower. Does it look anything like a flower? No. But this is a flower because it contains the male and female part. So it has to be called as a flower. Now, when you closely look at the left picture, you would find a spike-like thing. These spike-like things basically are representing the non-flowering bracts. Non-flowering bracts. This is, or maybe this is, these are non-flowering bracts. They are covering the flower, or you can also say they are covering the flower bud. And now they need to open. They need to open. Remember, non-flowering bracts need to open. When they open, how do they open? Let's go back to the previous slide. They would be opened by open in the season. In the season, the lodicules would push from inside to open the flower, to open the flower. So these lodicules basically are pushing, so the non-flowering bracts would open and the flowering bracts would also open. Now you can see inside the flowering bracts you would find these are the anthers. These are the anthers, which means they would be releasing the pollens. And also, these are also anthers, but one part here is the stigma. These brushy parts are stigma, and they are basically there to and trap the pollen from wind. So they are high in the air. They are high in the air, and this is the brushy stigma you can see. Brushy stigma. These are the structures of the. This is the structure, and there is another particular word. The anther is pendulous. Pendulous basically means hanging in the air. Hanging in the air. Why? Because the wind would reach here. The wind would take the pollens. This is the wind, and the pollens are here. So the pollens would be taken away. Pollens would be taken away from the pendulous anther. All right.
Yeah.
Any problem till here?
No.
Okay. Now this is a table you would be given in this particular slide. Also, you may find it in your book. Um, you would find the different characteristics and different differences in the insect and wind pollinated flower. Okay. So next come the fertilization. See, once the pollination is done, what was the pollination? The falling of the pollen on the stigma. Once the pollination is done, now the next step comes, and that is the fertilization. The fertilization is fusion of sperms and eggs sperm and egg to produce zygote. To produce zygote. So you can see the pollen is falling on the stigma and the tube is made. You see, this is called as the pollen tube. It is using the sugar, performing the respiration. The pollen tube would be grown. Okay, remember, the pollen tube would not enter straight into the ovule. Why? Egg is not closer, or you can say closely placed here. The pollen tube would enter from the region where egg is closely placed, and egg is closely placed here. So the pollen tube would take a bypass from here, and it would insert into the ovule. It would insert into the ovule through a part that basically is called as a micropyle. Micropyle is the same part from where the water is soaked. Water is soaked when the seed is sown. So the remember, the pollen tube is being made. The pollen tube is being made by the pollen tube nucleus. Pollen tube nucleus. And the generative nucleus, please look closely at the pollen to find the references. The generative nucleus divides into two sperms once it reaches close to the egg. So you can see now here you can find male gametes or sperms. So pollen tube is being made by the pollen tube nucleus, but the generative nucleus would not divide until it reached closer to the egg. What is the benefit of it? Basically, the sperms are very fragile, so they are not made earlier. They would be made when the generative nucleus is sufficiently closer to the egg. It would be divided. Now you have to focus on one more thing. Once the sperms are made, one sperm would fertilize the egg and the other sperm would fertilize the definitive nucleus. Now, as I have said, the sperm plus egg, they would be making the zygote. But the second sperm plus definitive nucleus that was placed in the ovary. It was placed in the ovary. When they would combine, and this does not mean two sperms, it means second sperm. They would make endosperm. Endo means inside, and sperm means basically the seed. Endosperm means food inside the seed. Remember that the definitive nucleus is responsible for making the food inside the seed. So when the second sperm combines with the definitive nucleus, it would be it would be making the endosperm. It would be making the endosperm, and this phenomena is called as double fertilization. Why do we call it double fertilization? Because two sperms are joining two nuclei. One is egg and other is definitive within an organ. So we name it double fertilization, and it has very important significant, and that is it ensures that the seed would grow by storing the food into it. It would make sure that the seed would survive and it would grow. Seed would grow by storing the food into it, by storing the endosperm into it. So always remember, suppose when you talk about a walnut, or when you talk about a peanut, when you talk about an almond, what these are basically, remember, these are not dry fruits. These are seed foods or endosperms that you are eating. All these are endosperms. Fruits. Fruits are, I would never say that the fruits are anything like a dry fruit or seeds. The walnut, peanut, and almond and cashew nuts, everything, these are seed foods. These are not fruits. A fruit is anything, this is also need to be defined. A fruit is anything made by the flower, made by the flower. It could be a tomato, it could be a tomato, it could be a bitter gourd, it could be a bottle gourd, ladyfinger, made by the flower. Okay. It could be an apple. Anything made by the flower is called as the fruit. There is no such thing as vegetables in biology. Anything made inside, I have already told you that anything that is made underground, that is the root, and anything that is made above the ground by the flower is a fruit. There is no such thing as vegetables in the biology. So you got the idea. This was very typical, very technical topic in which we understood why the pollen tube is penetrating into the ovule closer to the egg. Second, we said that the sperm would not be made by the generative nucleus the until the generative nucleus is closer enough to the egg. Third, two sperms are made because one sperm is meant to fertilize the egg and the other sperm is meant to fertilize the definitive nucleus. Definitive nucleus would be making the endosperm, and endosperm is the food of the seed. Food of the seed. Now, another important information, once the fertilization is done, fertilization is done. Now, ovary would start turning into what? Can you answer this? It would start turning into dash, and the ovule would turn into dash. In what thing the ovary would convert?
What do you mean? Like,
No, I told you that once the fertilization is done, the ovary would turn into
yeah,
the fruit. And
yeah.
what about ovule?
The ovule would turn, uh, would it turn into the seed?
Yes. Just, just like call your imagination. See, suppose this larger circle is ovary, the smaller circle is the ovule, and this even smaller circle is the egg. Now once the fertilization is done, the egg would now become a zygote. The the ovary would turn into a fruit. And what is present inside the fruit? Zygote. Does the zygote reside directly into the fruit? No, it is covered by the seed. So step by step you can understand, if egg into zygote, then ovule into seed, and then ovary into fruit. So fruit is containing the seed which is containing the zygote. Understood?
Yes.
Okay. Now once the fertilization is done, I have already told you ovary is turning into fruit and the ovule is turning into seeds here. Many types of seeds. Now, okay, now this is something very important to understand here. Structure of a seed. Okay, see, the seed is a baby of a plant. Be careful while listening to my question I'm about to ask. When the human baby is given birth, does it develop a new organ after being born?
No.
No. What happens? It has developed everything inside the mother and then it would just be born. Similarly, when the seed is there, the seed would have its own uh, root, basically called as radical, own root, stem, which is called as, or you can say shoot, or you can also say gonna be shoot, which is called as plumule, and leaves, which are called as cotyledons. This is very easy to understand if we make an analogy with the human baby. It contains mostly, if it is a normal baby, it would be containing all the organs, and it would just grow those organs once the baby is born. So it is a very typical concept. Every time when we look at the seed, always remember the root would come out of the point that is called as radical. It would turn into large root when it is sown. Plumule is the point that is supposed to be moving above ground. It means it is going to be the shoot moving above ground. Now, the cotyledons. These are cotyledons, this one and this one. These leaves, they have a feature that basically disturbs many students. Leaves surrounds all the endosperm. One thing that you need to be understanding very clearly, that the leaves of the seed are colorless. Leaves of the seeds are colorless. Why? Because there is no light. When there is no light, the seeds are the seeds are not going to develop the chlorophyll or chloroplast. They would only turn green. The seed would turn green when they would be, sorry, the cotyledons, the cotyledons would turn green when they are exposed to light. through light. So another very important thing, leaves surround the endosperms. Leaves surround the endosperms, and if there are two leaves, okay, I need to rewrite this sentence. Leaves surround the endosperm. If there are two leaves, they would totally the endosperm, cover the endosperm, and seed would be called as uh, dicotyledon. Dicotyledon, or one more thing, or maybe non-endospermic seed. You know why non-endospermic? Because you cannot see the endosperm. The endosperm is there. The endosperm is there, but you call it non-endospermic because the leaves have completely surrounded the endosperm. The cotyledons have completely surrounded. Only when they are two. This is very important to mention here. Only when there are two leaves, they would totally cover the endosperm, and seed would be called as dicotyledonous or non-endospermic. But if the leaf is only one, seed would be named as monocotyledon, named as monocotyledon, ledonous, or endospermic seed. Endospermic seed. So you can see here, I would advise you not to read. I added this table here, but these tables are not required. I will send you this annotated slide. So just ignore these. Just remember the definitions that I wrote here for for dicotyledonous and monocotyledonous. If you see here, this is a bean seed. This is a bean seed. Look at this. This bean seed is containing two cotyledon, and you cannot find the endosperm in here. So that is why it is a dicotyledonous seed. But when you see a corn seed, a corn is containing one cotyledon, so it would be called as the monocotyledonous, and you can also see the endosperm. So this is the basic difference. If there are two leaves, they would surround the whole endospermic tissue. If there is one leaf only, it would not be completely surrounding the endosperm tissue, and you can see the endospermic tissue in there. So, but it is a reality that both do contain endosperm, endosperm. Only it is hidden. It is hidden in dicotyledonous seed. Cotyledonous. Got it?
Yeah.
Now, what is an endosperm? Remember that an endosperm basically, typically, is a, what I must say, it is a collection of food molecules containing fats, carbohydrates, and proteins. Once sown, once seed is sown, the seed enzymes activate breaking or digesting the stored food to use it for growth. Use it for growth. You can see here to use it for growth. Okay. Um, I would teach you the dispersal later. Let's see the germination of seed. The germination of seed is when the seed reaches a spot, favorable spot, and uh, plans to grow. Remember that the conditions for germination, by condition of germination, I mean what are the minimal requirements for the growth? Number one, suitable temperature. Number two, enough water. And number three, oxygen. Oxygen is required for respiration. The suitable temperature is required for the activity of enzymes. And enough water is to activate the enzymes, because always remember that the enzymes work only in aqueous environment. Only in aqueous environment. So once the seed soaks the water, all the enzymes would be activated. You would see the amylase to break the starch and the maltase to break the maltose. The fats are there, so the lipase would be breaking them into fatty acids and glycerol, and the proteins are there, so the proteases would be breaking them into the amino acids. So that is how, and always remember, you must not forget the minimal conditions required. Must not. It's a very important question. Oxygen, water, and the temperature. Suitable temperature must be there, enough water must be there, and oxygen must be available. Is that clear?
Yeah, so oxygen has to be available, right?
Yeah, oxygen must be available.
Okay.
So we were talking. Okay, these are the types of seed. Okay, now the two investigation you will find here. One is very simple, and I guess it's also have been excluded. You have to examine the dicotyledonous seed basically. That is very rare to appear in the exam, and that is excluded as well. You don't have to worry about this investigation. This is an important investigation. I'm about to scroll toward. This is an experiment in which you have 4 test tubes. Every test tube is containing seeds. Every test tube is containing seeds. The cotton wool, the C test tube is containing the oil above it and the boiled water. And the D test tube is also containing wet cotton wool and seeds, but it is being kept at 4 degrees. It is being kept as 4 degrees Celsius. Now the question is, in which one of out of all of them, which one is most suitable for the seed germination?
Um, the A,
What's your logic behind it?
because 20 is like near room temperature, and
Okay.
so that's optimal for growth.
Hmm.
There's wet cotton wool, so there's water available. There's oxygen
Okay.
available.
So water available and oxygen is also available.
Yeah.
What about, so your answer I should mark, I would circle the A. Now, Why is there no seed germination? Because it is dry. So no water.
Yeah, no water.
Okay. Why would it not occur here?
Because it's oil.
Oil.
So
It
there's
would
very less oxygen.
uh, not let the oxygen dissolve into water. Okay. What about the boiled water? Is there any significance of it?
Um, doesn't help with the enzymes because it's gonna kill the enzymes.
No, 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 no. It you can also say that, but basically boiling leads to release of gases from water. From water. So if there was any oxygen in water, it would be released. Got it?
Yeah,
So no growth. D, the temperature is not suitable. So your answer is correct. All right. So the last topic is remaining, that is seed dispersal. Very easy topic.
I have my physics class like starting right now.
Yeah, okay. All right, no issue. Thank you.
Um,